All right, so do you guys have questions before we get started? I know there's a way to turn off just that one. Okay. You guys have any questions about mitosis, meiosis, biology in general, Dr. Pogorski, his testing style? All right. This is what taking learning into your own hands looks like, right? Did I see a hand?
What are the chapters that are going to be on the test?
If I remember right, you guys can correct me if I get it wrong, I think it's from like oxidative phosphorylation on, right? Or was oxidative phosphorylation on exam 1? It was not? So it should be oxidative phosphorylation and on. I don't know the chapter numbers that line up. I think that's like 9. I don't know the chapter numbers. But yeah, oxidative phosphorylation on is exam 2, which opens on next Tuesday, by the way. So super sessions are coming up. Mine will be Wednesday. Hillary's should be Thursday, and then Chase, the UTF, will probably do one Monday. Go to all of them if you can, honestly. This is one out of the 4. This is at least the harder 2 of, it's the top 2 of 4, I guess you could say. I don't know how you say it. But and it depends. Maybe you're like genetics isn't hard at all. Then we all hate you. But you'll breeze through it. But this is one of the harder ones, so study it. All right, so here's some, technically not memes, but tweets that I get a kick out of. One of them's from like 2017, so it's super old, right? Jay Cutler passed his physical and is officially a Dolphin. If football didn't exist, this would be a way cooler tweet. And then doctor looking at my x-rays. Doctor, this is exactly what I was afraid of. Me, what? Doctor, skeletons. Because it's October. Skeletons. It's gonna be a spooky one, guys. All right. Fun, technically not biology, but physics fact for you. I'm pretty sure that's pronounced Betelgeuse. I don't know. Would explode, transiting from red supergiant to supernova. So basically, if this star, I don't know how far away, that's like different galaxy, right? Exploded, our sky would light up for 2 months. Like it would look like daytime for 2 months straight, like biblical stuff, right? And they don't know when this is going to happen. It literally could happen tonight. It would just explode, and it would look like the sun was up for 2 months. There'd just be no nighttime, which would actually not be great. You're probably thinking, great, I can not get tired. I can stay out 24 hours. But then plants would not stop photosynthesis and wouldn't know when to start again, and all kinds of things would go bad. So it's actually not great. But I don't think anyone would die. All right, study tip is to pick a specific time and a specific place to study a specific topic. Okay? They've done studies that the can't think of the word that I'm trying to. The situation you put yourself in while learning, if you're in that same situation while testing, you'll do better. So if you sit on your couch in just your boxers and you got like Metallica playing and you're eating Cheetos with one hand and like kind of scrolling through the slides with the other hand, hopefully that's not what you'll do in the testing center. You're not allowed to. But there will be sort of a disparity between the way you learned and the way you tested. So I'm not saying like, get like 40 people together and sit in a room with like TVs showing nature scenes for no reason and the clock that no one actually looks at. But just, say, okay, I'm gonna sit down and I'm gonna study biology at 5 o'clock for an hour. I'm going to do it in my bedroom, in the living room, in the library, in this building, wherever, and at least try to make it somewhat formal so that when you go to sit down and test, which is also a somewhat formal circumstance, you're taking it more seriously. And your studying will go a lot better if you're wearing pants. It's just, that's also been studied. I don't know. Probably. I feel confident that you study better with pants on. All right. Before we jump into material, I know I already asked, but any questions? Again, we have lots of open time, so ask away. Plus, for a lot of you, and for me when I first took 1610, I'd never studied mitosis and meiosis before. I hadn't even heard the words. Okay, so do not feel like you're the only person in the room who doesn't know what's going on. Sometimes I still don't know what's going on, so we're all in this together. All right, so, oh yeah.
Sorry, so maybe you'll go through this today, so just tell me real quick. Can you go through the stages of mitosis?
Yes, definitely. Mm-hmm.
Okay.
Other questions? All right. So again, I take Dr. Pogorski's slides word for word, and I put them up here so that we can all learn to decipher his like Egyptian who knows what. He has a very formal way of talking about biology because he has a PhD in genetics, and he's been doing this since before we were born. So we have to learn to break his stuff down. Okay, so it says mitosis is the garden variety form of cell division that conserves chromosome number and genetic information between mother and daughter cells. And you go, I don't really know what any of that meant. So first of all, what's mitosis? Like the most general term you can give.
Division of the cell.
Division of the cell to get daughter cells with identical information, same number and same type of chromosome, right? So mitosis is just cell division in regards to genetics, and passing on that information. And they pass on other stuff, but we look at chromosomes. So it conserves chromosome number. What does that mean? Same number of chromosomes. How's that possible though? We duplicate and we cut in half and we do all this stuff. How does it end up with the same number of chromosomes? Okay, so we make a second DNA strand, but now we have twice as many chromosomes, don't we?
No, it's still just one.
No, it's still one chromosome, okay? But then we cut that chromosome in half, so don't we have half as many chromosomes?
It's called chromatid.
We have half as many chromatids. We still have the same number of chromosomes, okay? And we'll get into the details of why, but just for right now, just know mitosis starts and ends with the same number of chromosomes per cell, right? Obviously, we're ending up with more and more and more chromosomes and more DNA, because that's how you go from a fetus to an adult. You got to get more chromosomes, but per cell, same number, right? And genetic information. What does that mean? It conserves genetic information. Okay, so it conserves DNA, but what does that mean? Conserves DNA. Same DNA, right? We're not changing what kind of DNA is in there. We're not changing sequences. We're not changing nothing, right? There's no mutations. There's nothing. We're getting the same product as what we started with, okay? Between mother and daughter cells just means, the mother cell is the one that splits into two daughter cells, right? Meiosis is the division of sexual reproduction. So this is sperm and eggs in humans, right? That reduces chromosome number and creates genetically diverse daughter cells. We haven't gone deep into meiosis yet, so don't panic on that. Just know that meiosis reduces chromosome number and you get genetically diverse daughter cells. That's why we don't all look the same, right? Because every sperm and every egg is slightly different. Even from one parent, one mother has thousands of eggs, and each of those eggs is slightly different from the other. So that's why you don't even look, you look alike with your siblings, but you don't look identical. Unless you're identical twins, because you came from the same egg. So that's a little bit obvious. Okay, questions just with definition. It's always where you got to start. Define the vocab.
What does the garden variety form of a cell mean?
It means that, I don't know the percentage, but the vast majority of cell divisions are mitotic, not meiotic. So almost all cell divisions are going to be via mitosis. Only egg and sperm do meiosis. And considering how many cells you have in your entire body, egg and sperm make up just a small portion of that. Okay, other questions? All righty then. All right, so again, more definition. Chromosomes are carriers of genes, right? They're that famous sort of X-shaped looking thing that we've all seen before, especially if you were in class today. But they're the ones actually carrying your genes that are in the chromosome, okay? Mitosis acts on duplicated chromosomes to ensure an even division of chromosomes and therefore genes into daughter nuclei. Somebody want to tell me what the heck that's supposed to mean in normal words?
So you start and end with the same amount of chromosomes. That's what mitosis does. It divides the cells, and so we have the same amount.
Yep, you duplicate the genetic information, but then you cut it in half and split it, and so you started with 1, you turned it into 2 per se, and then you split that in half between 2, and so now each one has 1, right? Basic algebra. So because we have evenly divided the chromosomes, we have evenly divided the genes, right? If chromosomes carry genes and you split chromosomes evenly, you have also split genes evenly. And that's how you don't end up with difference in genetic information. Okay, you split it right in half, and they're identical. You can think of chromosomes as sort of the vehicle for the transport of genes, and we'll get more into chromosome structure in a little bit, but it's basically just proteins and nucleotides, right? And nucleotides are monomers of what? Nucleic acids, which is a whole bunch of nucleic acids is DNA, right? So it's just DNA and proteins all kind of packaged into that classic X shape, right? Actually, they spend most of their time just floating around. But when we can actually see them, they look like that sort of X shape. Do you have a question?
So like map like DNA, and why does it always show like the chromosomes and like the X's, it's just one?
That, and I think that's actually the next slide. So you're asking why is it X-shaped if it's only one chromosome?
Yeah.
So why is there a, where's my pen? How's there a left side and a right side, but there's only one chromosome? See, that's the trickiest, I would say that's the trickiest part for most students is going, but that's 2 pieces. Because we started with just this, and that's one chromosome, and then we duplicated it, that's still one chromosome. It's because chromosome technically is also, the definition is carrier of unique genetic information. So this is one chromosome because it's unique to everything else, right? It contains its own unique DNA to everything else. But if we duplicate it exactly, these are actually identical to each other, so they're considered just 2 pieces of the same chromosome, right? I guess you can compare it to like tires on a bike, right? Each one's a tire on its own, but they're exactly the same, and they're in that sense one bike, right? I know it's not a perfect analogy, but each of these is exactly the same as the other, so they're still just considered one single chromosome. Does that make sense? So it makes that X shape, and it has 2 pieces, but the 2 pieces are identical, so they're considered one, especially because they are physically attached to each other, right? So they're one whole unit made up of 2 identical pieces, right? I know that's tricky because two tires doesn't equal a bike, but two chromatids does make a chromosome. It's tricky.
So then what do we say we have like 20X pairs of chromosomes?
Because we have 23 pairs. So I wish I had a picture of it. Your DNA is made up of 23 pairs of chromosomes. If you were to split each of those in half, you'd have 46 pieces, but it's 23 pairs, like shoes, right?
So
I might have,
we're talking about a pair, it's not that, it's something else.
no, so this is a pair,
A pair.
but it's one chromosome. It's the worst, guys, I know. You could think of them as like if you had 2 left shoes, I guess, but they're the exact same shoe, and somebody asked you for, like, okay, so I have 2 of this shoe, and somebody said, I want that shoe, like the right gray shoe. I could give them either one, and I'd be giving them the same shoe, right? And if you put them next to each other, they're still the same shoe. And I don't know if you somehow like stitched them together. They would just be one shoe because you'd wear it as like a single shoe, right? It doesn't have to make logical sense. Just know, each piece is a chromosome on its own, but when they're together, they're just like a joint chromosome, right?
So could you like say that there are 46 chromatids, and that'd be technically correct? Like 46 or 22 pairs?
23 pairs of 46 chromatids, yeah.
All right. Okay.
So we start using the term chromatids when there's two. After we start with a chromosome, right? First a little bit.
Yeah.
Go to chromatid when there's two.
Yeah.
Then they become chromosome because you get split.
Each one's a chromosome separately, but if they're identical to each other, they're sister chromatids and one chromosome. It's the worst, guys. This is what takes like the longest to wrap your head around, and to be honest, I don't have a perfect analogy for it because there really isn't. It's kind of its own deal, but one chromatid plus one chromatid equals one chromosome. Okay, it's the worst.
What's the difference between chromatin and a chromatid?
Chromatin's what it's made of. Chromatin is that mix of proteins and nucleic acids all jumbled together. When you take all of the chromatin as a whole, it's a chromatid. And I think there's we might have a slide on that in a second here.
Can we go over that wording, like chromosome?
Yeah, I think I've got a slide on that. If not, I'll take a second to explain it. I can't remember what slides I do or don't have. You guys loving it? Who's still not really getting the whole 1 + 1 = 1 thing? No one wants to raise their hand because you don't want to look dumb? What if I raise my hand? I don't get it either. I don't know. Figure it out on your own. 1 + 1 = 1 with chromosomes. I hate to say it. Now, if we had a different chromosome, this is going to make it worse. These are 2 chromosomes, okay? Because they're physically separate from each other and not genetically identical to each other, okay? Because this one birthed this one, and this one birthed this one, but these did not birth each other, okay?
So basically the chromosome is exactly the same part of it, right? One of those chromatids is the same exact thing as the other one, right?
Yeah, they're identical halves of each other.
So that makes sense that they'd be just one thing because they're one together.
Yeah, they're the exact same thing as each other, so even if you looked at them separately, they're identical to each other. So they're going to do the same thing, right? If that left one, for whatever reason, is for blue eyes, which that's absolutely not how this works, but run with me. If the left half was for blue eyes, the right half is also for blue eyes. So either gene that I get, I'm getting blue eyes, right? That's why they're just one chromosome. They do the same thing, right? Yes? No? Hopefully. Okay, we're going to actually look at the myriad of words on the slide now. So chromatin is the assembly of proteins and DNA, right? So these are your A's, T's, C's, and G's that you've all seen. And then just regular old proteins, right? Mixed together. And they wrap around each other, and they wrap around each other over and over and over until they spiral up into that actual chromosome shape, okay? So chromatin is proteins plus DNA, right? Does that make sense? I will wait for acknowledgement.
Yeah.
Thank you. No one looks. Everybody's like, I don't know. All right. So they make up a chromosome, right? A chromatid of a duplicated chromosome is one molecule of DNA, right? All of that is one molecule of DNA, okay? But each of those is a separate chromatid. So together, right? Each chromosome together. Sorry, I'm saying this wrong. I made it worse, didn't I? Each chromatid is one molecule. So that's one molecule, that's one molecule. One chromosome, two chromatids, or two molecules. You can think about it either way.
So two things of DNA.
Two molecules of DNA,
Uh-huh.
which is the same thing as two chromatids, because a chromatid is a molecule of DNA, equals one chromosome. But also, if they're separate from each other, they also equal one chromosome. Just trying to remind you guys that you hate biology. I just don't want you guys to forget that one chromatid on its own is one molecule of DNA, is one chromosome. Two chromatids is two molecules of DNA, but it's still one chromosome. It doesn't have to make sense logically. You just have to memorize that that's how it works. Okay? So again, two DNA molecules, 1 and 2, in a duplicated chromosome. This means it's identical to its halves, right? Mirror images of each other is a single, no, they derive from a replication of a single DNA. So this one replicates, and that's how we get that one. Okay? Left one gets duplicated, we get the right one. So they're sister chromatids. That's where this vocab word comes from. They are identical to each other. Okay? Again, a chromosome may contain one or two DNA molecules, one or two sister chromatids. Well, I guess if it's one, it's not really a sister chromatid because it's not a sister to anything. One or two chromatids, and they're one chromosome, regardless of one or two chromatids. Three chromatids is a different thing. That's a genetic disease. I don't think we cover that for a little bit here. So, questions? Can we stop? We're done. Go home. Drop out. No, that would be a bad idea. I can't encourage that. It just takes a bit to wrap your head around a sort of unique concept. Not a lot in nature works as a 1 + 1 = 1 thing. And for most of us, this is probably the first time we've heard any of these things, at least to this level, if not at all, right? Questions?
So if there's still just one molecule of DNA, it's still just a single chromatid, that's what it's called. And then if there's two, it's still just a chromatid.
Sister chromatids, if they're identical to each
They're
other.
identical.
Mhm.
Okay.
A lot of vocab. So maybe make a flashcard with chromatin, chromatid, sister chromatids, chromosome, so you can keep each definition separate from the other, right?
So if you have like two chromatids, they're like identical twins.
If they're sister chromatids, they're identical twin sisters, basically. If they're not considered sister chromatids, it's because they're not identical to each other. And for the purposes of this class, they have no relationship. So we don't really say like non-sister chromatids because we don't deal with that. Be grateful. Some of you will go on to take genetics, and it's a hoot. Yeah. Questions? Okay. Chromosomes are duplicated before mitosis. Mitosis is not the duplication of chromosomes. Mitosis is the division of cells containing duplicated chromosomes. Anybody remember when chromosomes are duplicated? Interphase. Anybody remember which specific piece of interphase? S phase. S stands for synthesis. You can think to synthesize means to like make more. We're synthesizing duplicated chromosomes. Okay? So duplication, this part is interphase, specifically the S portion. This splitting, that's mitosis, right? Everybody feeling that? We have one chromosome. We undergo S phase. We have one chromosome, but with two identical sister chromatids, then we split them into separate cells. How many chromosomes do I have here? How many chromosomes do I have here? How many chromosomes do I have here? 2. But didn't I say that we conserve chromosome number? 1 and 2 are not the same number. Per cell, right? So we still ended up with 1 per cell. That's the, some people get tripped up on that. They go 1, but still 1, but now 2. That's not conservation. It is per cell. Okay? And again, this is 1 chromosome. But somehow you split 1 in half and you get 2, not 1/2. So it doesn't follow like arithmetic rules. It follows biology rules, which has no rules. It's like English. Questions on just this? Questions on, oh.
What was interphase again?
Interphase is basically everything that's not mitosis. It's when mitosis ends, you go back into interphase until you start mitosis again. But it has some phases, and we'll look at that in a little bit. And I think we get into deeper detail next couple of lectures here. But it's just when you're not in mitosis, you're in interphase. Questions on anything before this, except for the 1 + 1 = 2 thing, or 1 + 1 = 1 thing. All right. I just don't want to talk about it anymore. I'm tired of it. Very important, right? I took like 5 minutes to animate those stars so that you guys would remember. 1 chromosome. If that's all you take out of this SI tonight, I'll be a tiny bit disappointed because it was 50 minutes long, but it's the most important, most difficult, I think, to get our heads around. Okay?
So you're saying in step 2, 1 chromosome.
That is 1 chromosome. That is 1 chromosome. That is 1 chromosome. That is 1 chromosome. And these cells contain exactly the same genetic information as each other. Is everybody feeling that?
So I'm in like step 3. So if it's the same chromosome being separated, is it still just 1 chromosome, or is it 2 at that point because it's in 2 different cells?
It is now 2 separate chromosomes. It's 1 chromosome per cell. So we ended up, yes, with 2 chromosomes, but we consider it conservation of chromosome number because we look at it per cell. That makes sense? So we get 1 chromosome per cell. We get 2 cells, but it's 1 per. Other questions? We're doing all right on time.
So you only get 2 chromosomes from each time, right? You can't get like multiple.
Well, I mean, you're taking every chromosome in the cell and splitting it, but you get
cells from this whole process.
Yeah, so you split that and you get 2 cells. You don't split it into like 19 cells. It'd be faster, but you'd also have to make the cell 19 times larger to accommodate 19 times as many chromosomes, and then so we duplicate each chromosome and split each chromosome in half, so you end up with 2 cells. Yeah, but you're splitting up to thousands of chromosomes. Just depends.
So then in step 3, do those 2 separate cells have 1 chromatid?
Yes. 1 chromatid, which is 1 chromosome. And then when those duplicate again, it's 2 chromatids, 1 chromosome. We're starting to get there. I can feel people kind of like nodding their heads a little bit. It takes just a while to wrap your head around this is not real math. Then again, once you get past algebra, is it really real math? I took calculus, and I was like, this is dumb. I don't need this. All right, I'm going to medical school. We don't do calculus. We really don't, though. That's the funny part. Mitosis separates sister chromatids so that each daughter nucleus receives one of the 2 identical DNA molecules. Somebody break it down for me. Regular words.
So in step 2, you have the 2 sister chromatids with identical DNA, and then mitosis is just separating those into 2 cells. So they each get the same package of DNA that's identical.
Mm-hmm. Who feels like that's the 10th time we've said that today? I do. It's because, honestly, the more times you say it and hear it and draw it and read it and hear it and say it and draw it, the more it makes sense, the more you really remember it. Because right now you may go, oh, like I get what we're talking about. But a week from now, when you've studied 15 other topics for just this class and then 30 other topics for all your other classes, you know, you may have pushed this out of your head. So we're going to say it over and over, and I hope this isn't the last time that you look at this. Do it over and over and over until it makes sense, but you know that you're not going to forget it a week from now. And don't forget, the final's cumulative. You have to remember this in December. So good luck. I only have one cumulative final this semester. But also for my Spanish class, the 4th test and the final are the same week, which is really annoying. So this is what I was talking about when I said we would look at what interphase is. So interphase is all of this, I want to say blue-green arrow. Is it blue-green? Yes, frick yeah. I never get those right. Interphase, right? Made up of 3 parts, G1, S, G2. We'll go into detail further of what exactly goes down in those. S stands for synthesis. G, I want to say stands for growth, but it doesn't sound super scientific, so I'm probably not right. But, what? Gap?
Yeah.
All right. But I call it growth because that's what actually happens, right? The cell physically gets larger. The number of lysosomes increases. The amount of ER increases. Everything gets bigger, right, in both of these phases. We'll get into the details of exactly what happens in exactly which G, right? But it's the growth phase. It might be the gap phase. I call it the growth phase. Call it what you want. And then S phase is for synthesis, right? What's happening in the S phase? Chromosomes get duplicated. All right. Then we have the M phase, or mitotic phase, which is when mitosis occurs. But you may notice there's also a tiny little sliver for cytokinesis, because cytokinesis isn't technically mitosis, nor is it technically interphase, but we put it in the M phase because it always occurs immediately following mitosis, right? Once we finish all the phases of mitosis, you go straight into cytokinesis, but it's on technical definitions not the same thing. But we're going to look at the 6 phases, and the 6th one is cytokinesis. So if you want to be super formal, it's not part of mitosis, but it is. Questions?
So do prokaryotic cells do a lot of similar things?
Like, do they do mitosis in the same way?
No, just like the whole cycle as a whole, like before dividing and.
I want to say yes. In essence, it's the same process. They may have their small differences or whatever. I want to say yes. I don't know. And that's also probably not something you'd be tested on. But I think it is essentially the same process. Other questions? If I don't know it, it doesn't matter, right? That's what I'm saying. All right, so let's actually break these down. Oh, I love that. That drives my OCD up the wall. G2 of interphase. Cells just prepare for mitosis, right? So that's G2 is this, right? So not actually part of mitosis, but it's important to recognize that that is the step immediately preceding mitosis, if that makes sense. So cells just get ready, right? Oh, how did it switch to the... You'd think at 22 years old I wouldn't already be like an old person with computers. Cells prepare for mitosis, right? Prophase, so this is where he had a mnemonic. Do you guys remember his mnemonic? Purple peas make awful tarts. And you can come up with your own mnemonic. I don't want to because I guarantee I'll say something inappropriate accidentally. So just make up your own mnemonic or use purple peas make awful tarts, right? But those are essentially the 5 stages. Just don't forget that G phase leads into prophase, right? So prophase, the chromosomes actually become visible. Does that mean chromosomes are invisible? Yes, no. They're so loosely packed, so just open and out in the cell that you basically can't see them even under a microscope, right? You can kind of see like a little bit, but you couldn't say like that is definitely a chromosome, right? They're just like soup, right? All over the place. In prophase, they actually become visible and they make the chromosome shape, right? Nuclear envelope dissolves. So if this is our cell, or this is our nucleus, sorry, it starts to kind of break apart, right? Why the heck would I break the nucleus apart? Doesn't that seem kind of dangerous? We're exposing our DNA. Huh? Somebody said it. It wimped out. Who knows? Why would I break the nuclear envelope?
You have to do that to get to the DNA to copy it.
Yeah. How else am I supposed to send it from one cell to the other if it's trapped in the nucleus, right? Got to break it down. Otherwise, I can't reach it.
So it's like the whole nucleus just kind of disappears, or is it just?
Well, the nuclear envelope. So it's going to reform. It's just that layer breaks apart. Doesn't like dissolve or anything. It just breaks apart. Chromosomes leave. We do the whole process, and then it'll reform.
Is the envelope the same thing as the membrane?
Basically, yeah. Other questions? All right. Pro-metaphase. Okay. So it's prophase, then metaphase. So kind of that in-between phase is pro-metaphase. So even if you can't remember pro-metaphase, you can remember, well, it's that phase between pro and meta, so it's pro-metaphase. We separate into two identical daughter cells, and the nuclear envelope breaks down. Did this not transition over right? I think it's supposed to mean chromosomes. Anyway, it's the phase between prophase and metaphase, and the nuclear envelope's like all the way gone. Not like disappeared, but it's out of the way, 100%. Prophase, it's just kind of starting to break down and fall apart, but pro-metaphase, they're just, it's gone, right? It's completely out of the way. So that's what this is what actual drawings of it look like, right? Well, so this is a drawing. This is an actual light microscope image. It's been stained, right? You don't have super cool looking supernova DNA. It doesn't actually look like that. But if you don't stain it, it's really hard to tell what's going on. So G2, we have duplicated our chromosomes, and we're ready to go, right? Prophase, they actually form that classic chromosome shape, right? The classic sort of X-looking thing, right? That's just prophase. And again, notice how there's some breakdown of the envelope. Then pro-metaphase, the envelope is like all the way broken down, right? And out of the way. And these microtubules are starting to reach out and actually connect to the chromosome. What's that part where it connects called? The region's called a centromere, that whole middle region, but what's the specific part where it attaches? Kinetochore. You can think kinetochore is where it connects. They kind of sound similar, right? So the centromere is this whole central region, but that specific black dot, I guess you could say, that specific spot where the microtubule attaches, that's the kinetochore.
This happens throughout this, like in the cell, like general cell.
Yeah, so this would be like a whole cell, and from the polar end of one cell and the polar end of another cell, it reaches all the way across. Basically in this middle region, yeah. The chromosomes hang out in the middle. The kinetochores reach out from each opposite end and grab on.
What happens to the endoplasmic reticulum during all this stuff?
Not much, really. Just hangs out on the side and gets ready to get shipped to whichever cell it gets assigned. So We duplicate everything, but we look at chromosomes because genetics doesn't really care about what happens to the ER. But there's like an ER over here and there's an ER over here, and eventually when we split the cell, otherwise you'd end up with one cell with a ER and one cell without, and then that one dies. So you do have to duplicate everything. We just care about chromosomes. Other questions? Last 3 stages. Metaphase, chromosomes become attached to the spindle fibers. Anaphase, they move away from one another. And then telophase and cytokinesis, and it looks like that last little bit didn't make it on there, but they move to the opposite ends, 2 nuclei are formed, cytoplasm splits into 2 separate cells. So I've done it this way in case you're a reading type learner, in case you're an auditory type learner, but I've also paired it with pictures. So if you're like, those words made no sense to me, let's look at what the actual picture is. The reason it's called metaphase, does anybody know what meta means? Middle, right? They line up in the middle on what's called the metaphase plate. It's an imaginary line. There's not actually like a plate in the middle of your cell, but they just call it metaphase plate, right? They should have just called it the metaphase line. I feel like that would have made way more sense. So these kinetochores have attached, right? And they're specifically lined up pretty much dead center, right? Anaphase, they actually pull apart from each other fairly violently, like they actually rip in half, okay? So they each get ripped in half and pulled toward one end of the cell or the other, anaphase. You can think of Annabelle, that creepy doll, right? I don't know. Did she ever rip anyone apart? I don't think she did, but she would, right? So just think of meta means middle, so they line up in the middle. Anna, like Annabelle, the creepy doll that rips people apart, who doesn't rip people apart, but she could. What if they don't split? Yeah, lots of stuff can happen where they don't split correctly. Down syndrome actually happens if you don't split the chromosomes correctly. You end up with an extra 21st chromosome. I want to say 20. It's called trisomy 21, it should be. So you end up with 3 chromosomes in your 21st pair. So it's not a pair, it's a triplet. And that's how you get Down syndrome. And lots of stuff can happen. There's trisomy, I want to say 18, and there's a couple of different trisomies. Yeah, so if they don't split right, you end up with lots of weird stuff. I think that gets covered more in like the genetics course.
So in the anaphase, what are those chromosomes at this point? Are they
Like this?
Yeah, exactly.
These are now each separate chromosomes, and they're also one chromatid. So this is one chromatid, this is one chromatid, this is one chromosome, one chromosome. It's the worst, I know. Chromatid is a singular piece, and if you put 2 of them together, you have sister chromatids, but you still have a chromosome because they're identical. You're going to have to hear it a million times before you're like, I guess I kind of get what that means. All right, telophase and cytokinesis. Does anybody know what telo means? Or tele, like telephone, television? Transfer or like long distance, right? So the telophase, the whole thing moves away. An entire cell, so all the DNA, all the lysosomes, all the Golgi, everything moves to one end or the other. They teleport, I don't know. But that's telophase. And then cytokinesis, they often kind of get grouped together because it sort of happens at the same time. As this goes here and as this goes there, we split the cell in half. Cyto, as in cytoplasm, right? What all the cells filled with. And kinesis as in movement or separation. So we're actually splitting the cytoplasm in half. Eventually that will completely split in half and you get 2 different cells. Does anybody know when they don't split like apart from each other? That when they separate but they don't physically separate? It's two different cells, but they're still attached. Did we not talk about this in class? Plants. So a plant cell duplicates, but never actually splits. This becomes cell wall. And then you build another one, and another one, and another one. So that's why if you look at plants under a microscope, they really are, every single cell is just stuck to each other. That's actually how they became called cells. The first thing they looked at was plant material. I can't remember which plant. Cork. And they saw this and they're like, it looks like little cells. Each individual square is a little cell. So that's why we started calling them cells. Because the plant cells separate, like they make two separate cells, but they don't physically leave each other. Animal cells, on the other hand, do physically split, right?
Is that what gives plants like the strong structures, that cell wall, and then division, basically just making more cell walls?
Yep. Exactly. You're just making it more and more rigid. The more of them you have, it's like more building blocks on top of each other. That's why it's a lot harder to push a tree over than a dog or something. Also, they're not rooted in the ground, but that's different. This is the fun part. Take a minute or two and figure out which stage each of those are. They're not in order, so you can't just go, oh yeah, purple peas make awful tarts, right? Go and look and say, okay, are they splitting? Is the nuclear envelope dissolving? Are the chromosomes getting split in half? Are they just lining up in the middle? Maybe talk to a neighbor, do whatever you got to do, figure out which phase is which. Are you what?
Including G2?
Yes. As far as I remember, G2 is part of this. All right. Let the voting begin. What do you guys want to say that first one, the top left is? Shout it out. Telophase. How'd you know? You can see this actual going each direction, and we're starting to come in on ourselves and eventually split, right? What's that second one then? This one. Metaphase. How'd you know that? They're pretty much lined up down the middle, right? What's the next one? G2. Why? Yeah, you can't see any chromosomes. Nothing's really happening, right? What's the next one? Anaphase. Why is that? They actually split from each other, right? Oh, that's prophase. What's the last one by logical deduction then? Prometaphase. So prophase, we can actually see the chromosomes as distinct figures. Prometaphase, they've actually broken out of the nuclear envelope, right? You guys actually did better than any other bio SI that I've done before. Most of the time, 2 people are anaphase, and they're wrong. So you should be proud of that. Also, if you can look at these and recognize the phases, you should feel very comfortable. That's as hard as it gets. Okay, so if you can recognize those, you're in good shape. And this is just a lot of definitions, a lot of vocab and structures. I'll go over it in my super session on Wednesday. And again, it's kind of just memorization stuff, so not the most complex thing in the world. All right, thanks for coming. Who's got my roll? Anybody not get the roll?
Let it go.
Come up and sign
Roll.
if you didn't get it.
Written.
You need to let it go, man.
You got something for me?
Let it go. What is? So like, we're talking about these.